Are you ready for something really weird? So we had talks about chemical life, and basically organic life still could be in different environment and based on different molecules, but it's still organic life. Now we go to truly inorganic life, and this is life in the computer. And it's my pleasure to introduce the next speaker, is Dr. Charles Ofria. He's a professor of computer science and engineering at Michigan State University. He's also president and one of the founders, as I understand, of the Artificial Life Society. Yep. President, yes. Yes, and it's a big community, I can tell you. Then he's the founder and a deputy director of the Beacon Center for Study of Evolution in Action. So he studies evolution in the computer and in the simulations, and he is one of the creators of the long-running software called AVIDA. This is artificial life simulations, and he studies evolution of digital organisms and digital evolution. So all those concepts we discussed in context of the chemistry can be now proven or disproven, let's say, or found in new concepts, I believe, with the help of simulations. Welcome.
Thank you, Svetlana. All right, so this will be interesting, giving the first of the talks of about the truly strange organisms. So in my case, I work with self-replicating computer programs, digital organisms, that will evolve inside of a computer and study evolutionary processes in that context. You can kind of think of these like very well-contained computer viruses that are subject to mutations that will actually change in interesting, meaningful ways over time. And I should note, I do most of this work under the Beacon Center now. So this is the Beacon Center for the Study of Evolution in Action, which really focuses on evolutionary processes in real time across all sorts of systems. So there are natural biological systems, both in nature and in the lab, but also engineered systems, so chemical systems, robotic systems, and of course, computational systems. And we use these systems both to understand how evolution works and to apply evolution to solve a whole range of problems. In order to talk about why artificial life systems are so useful for this, I like to use a quote from John Maynard Smith, who was a very prominent evolutionary theorist. So John Maynard Smith said, "So far, we have been able to study only one evolving system, and we cannot wait for interstellar flight to provide us with a second. If we want to discover generalizations about evolving systems, we will have to look at artificial ones." And I think a lot of people here would agree with the value of finding life in space, although interstellar is probably not necessary. My own motivation for getting into this area is a little bit different from this originally. So my own thought process to go this route actually started off with a keen interest in artificial intelligence. So in the early 90s, as an undergraduate, I was trying to learn everything I could about artificial intelligence. This is the direction I really had thought I was going to take my career at that point in time. But the more I learned about AI, the more I realized that real human-level intelligence was still 10 to 20 years away, just like it was in the 40s, just like it still is today, and the sliding window. And so I shifted gears to start thinking more about the process that created the one instance of intelligence that we know of, us. And that got me to learn a lot more about evolutionary biology. The more I learned about evolutionary biology, the more I became fascinated with the big open questions about how complex things arose, such as intelligence. But on the evolutionary biology side, these questions are very difficult to ask because when you're working with living systems, they reproduce slowly, so you have these long time scales you're dealing with, and they're messy, it's hard to collect all the data that you need. And so this actually led me to go back to the computational side to see how can I take my computational skills to study evolution better. All right, so to tell you about this, I'm actually going to jump back a little further in time to the computer game Core War. Core Wars. So Core Wars was a game where players are computer programmers that write these programs, put them in the core memory of the computer. The programs battle it out. Last program standing wins, right? And so it's a really fun, interesting game. And it turns out that one of the most successful strategies in Core Wars is to write a program that makes copies of itself. So when that original program dies out, you still have all these copies, all these clones of it to continue on the fight. But because it's in a computer, those copies are always perfect copies, and as such, there isn't any evolution in the system. So this leads a little bit later to a system called Tierra. Tierra was designed by a botanist by the name of Tom Ray, and he used a lot of ideas out of Core Wars, where he worked with self-replicating computer programs here. But put them in an environment where cosmic ray mutations rain down on them that trigger bit flips. And he started off this Tierra program with a simple self-replicating digital organism. It was 80 lines long, and let it fill up memory. What you see here is just a snapshot of the memory with a whole bunch of organisms in it. And he watched to see what would happen. All right. One of the things he saw that was pretty amazing was that these programs, over time, as they're making copies, were shrinking. They were getting shorter and shorter and more efficient. This alone would have been really fascinating evolution, right? But a lot more than that happened. One of the other things that he saw was that even though the fully working programs were able to shrink down to about 65 lines long, suddenly even shorter programs appeared that were about 45 lines long and started taking over memory. But they couldn't take over completely, and they would die out again, and then take over again and die out again, and they were oscillating back and forth. When he looked at these particular programs, what he saw was that they actually left out very important segments of their code and just executed their neighbor's code instead. So these were basically parasites that were using their neighbors in order to succeed. And there was a whole wide range of really interesting evolutionary dynamics that went on, all sorts of coevolutionary effects, even different forms of cooperation arose. And this really inspired me. And this led me to create my own system called Avita that was built to be an experimental test bed to study evolving systems, all right? And I tried to build it working with biologists in order to really be able to mimic the sorts of experiments done in bacterial systems primarily. So, for example, here's a couple of runs where what you'll see are two competing types of organisms spreading out over, these are in spatial worlds. Each dot there represents a full computer program. And on the left-hand side, it's obviously, they're competing in the environment that the yellow and green organisms evolved in. And on the right-hand side, they're competing in the environment the blue organisms evolved in. The big difference here is actually mutation rates. On the right-hand side, there was a much higher mutation rate. But this whole project was able to show that organisms would actually evolve robustness to mutations. And I could tell you a lot of really interesting things about that if we have time later. Now, in biology, it's very difficult to collect the level of data that we often need for things. And so this was just an example of a project looking at the tree of life for turtles that I found fascinating. But in order to get this level of data to build a phylogeny like this, what you need to do is survey what's out there and then extrapolate back to what must have existed in the past, right? And there's many very good ways of doing this. But we don't know every single organism that has ever existed along the line if we're working with these natural organisms. The big advantage of working with digital organisms is that we can save all of this information as evolution is progressing. So here's an example of a phylogeny from digital organisms. We have every single individual, every single genome. We know every mutation that occurred along the way. And in this particular case, these organisms were evolving in a heterogeneous environment. In the upper right corner there, you'll see the layout of the environment where there are various resources in different places. And we were actually able to further color the phylogeny based off of where each organism lived in that environment. So we can trace pathways through the world that this evolution occurred in. And this is actually just a piece of the full phylogeny, but it gives you the idea. All right. Now, once we have all this information, we can actually look at lineages for organisms over time. And so this is generation by generation. We can see every mutation that occurred, and we can start to analyze what's actually going on with these organisms. So if there are particular traits we're interested in, we can see that for that first trait, the first several organisms didn't have this particular trait. Then it evolved in, and then a few generations later it evolved away for some reason, then it evolved back, and we can start teasing apart what's going on there. For example, if this trait was valuable, why would it evolve away? Well, in this particular case, we can look at the other traits, and we can see that when that first trait was lost, another trait was gained, right? And if that other trait is more valuable, that's an easily worthwhile trade-off. And so often, traits are used as building blocks for more complex traits. The simpler ones evolve first, the more complex ones come later. And we actually did a much more controlled experiment looking at these dynamics. Here we have 3 different treatments that we used: a reward-all treatment, a control treatment, and an ecological treatment. In the reward-all treatment, we rewarded a whole bunch of different Boolean logic tasks that are of various difficulties for the organisms to perform, how many lines of code they need to do it, with the EQU equals at the bottom there being the most complex one. So our claim is that if building blocks are important, evolving all the earlier tasks would have been very important for evolving equals. So in a control treatment, we didn't reward the earlier tasks. We only evolved the complex trait equals. And then in the ecological treatment, we actually had resources associated with these different tasks. And when a resource is used, it's gone. And so this creates a nice ecological balance with the inflow and outflow of resources. Well, what happened with each of these? We ran 200 populations in each of these regimes. And what we saw in the reward all is about 1/3 of them actually evolved equals. In the control treatment, where they didn't have the building blocks, none of them evolved equals. So clearly those building blocks are important. What about the ecological treatment, where we don't provide them with as much resource, right? It's a limited resource. Well, it's interesting because there's not as much resource, so at that point you might think they do worse, but also there's going to be much higher diversity, because different organisms are going to start specializing on different traits, and trade-offs are much easier, because if you lose the ability to digest resources that everyone else is using, but gain the ability to use one that no one else is using, that's very valuable, so these trade-offs are easy. So in that last treatment, we actually see 167 out of the 200. So just showing that even such simple ecological dynamics added to the system turned out to be very valuable. Now, this is all looking at how does complexity arise in these digital organisms, right? Now, I'm interested in even much more complex forms of complexity, such as transitions in individuality, such as going from single-cellular organisms to complex multicellular organisms. How does all of that work? And this is a real fundamental question in evolutionary biology. So if we have ancestral cells, and let's say they're color-coded based off of what tasks they do, so these do the red task and blue task, whatever they might be. One of the things that we might see happen is that cells start grouping together. If by being in groups they're better at acquiring resources, or maybe they can escape a predator or something along those lines. There's lots of reasons for doing that. Once they're grouped together, they can start engaging in division of labor. So you have some organisms specializing in the red task, some specializing in the blue. Once you have that happening, you can actually start having more complex patterning, so developmental processes coming in. And in the digital organisms, we've been looking at each of these transitions and have been able to isolate individual transitions. And my big goal, what I really want to see, is a system where we set it up, and without ever influencing anything, we start with the evolution of single-cellular organisms, and over time we see them go through each of these transitions very naturally. And we now have enough where we can show each single transition that I think we're pretty close to this. So, for example, looking at the evolution of grouping, here's an example. On the left side, we have single-celled organisms that are digesting resources, and the resources are put together in such a way that when an individual identifies that they have a resource. They have different ways of notifying their neighbors so that those neighbors will be able to get the resource as well. As such, if they grow out to be multicellular organisms, they're able to get more of the resource, and that's what you're seeing on the right-hand side there. And we can even take some of these multicellular colonies, and this is actually from a different project where we fixed the colonies to have 25 cells in them in these 5x5 arrangements and made it so they had to do a range of different tasks. The ancestral colony was able to do one task and thus bring in one type of resource. And we saw generalists evolve pretty frequently, where each individual cell in the colony did the same thing, but they were each trying to bring in many different resources. But the most successful ones that we saw were evolved specialists, where even though every member of the colony was clonal, so they're all identical to each other, they came up with ways of specializing and dividing up the resources. Here's that specialist colony, and we can look at organism by organism in it to see how they take incoming messages to perform tasks, and they might perform a task themselves, but the partially processed results from that task they might pass on to another organism that can then do a different task and pass the results on to another organism to do yet another task. Right? So these organisms are all actually working together and communicating in order to successfully engage in multicellularity. And we can look at the entire groups here. Now, the next steps that I really want to take is make it so that people anywhere can use these systems really easily. And so these are actually videos from the web interface that I've built for this that I'm hoping to release in the next, I'm hoping for this summer, where anyone will be able to go on the web page and very simply run experiments. So on the left-hand side, you see them growing out just by a normal replication process. On the right side, they can actually bind to each other. This was actually set up like Will Radcliffe's snowflake yeast, using that same sort of setup that Lynn talked about earlier. And we see some very similar results there. We also already have an educational version of Avita Ed, which is available on the web at avitaed.msu.edu. If any of you want to check that out, it's actually being used in hundreds of classrooms around the country already. And this is a stripped down version of the basic Avita. So with that, if anyone has any questions, I think I have a couple minutes.
Hi, I saw from your graphs that a few evolved to do nothing. Why do you evolve to do nothing?
So, let me see if from these you mean? It all depends. In this case, I believe actually those cells never got full. So they can be as large as 5x5, but they don't have to fill in all of the different cells there. And there's a trade-off of how they spend their time, because if they're reproducing, they're not also pulling resources in. So not all of them would hit the balance of filling up the full, we call them multi-cells.
Morgan Cable, JPL. This is really cool stuff. In your simulations that now you're hoping by the summer to put online, have you thought about adding like a third dimension so things can grow in 3D? Or the more interesting thing I'd rather have you comment on is if you could introduce like surfaces and adhesion to surfaces or pockets and
yes.
Interaction with the environment.
Yes, so I actually think that those are some fascinating directions to go, and there's some really good physics engines out there. I was trying to keep it to 2 dimensions, primarily to keep it fast. So the videos I showed you were real time. I just in the web browser, so that's how fast they will ideally go. And I worry as we move to more dimensions, it's going to slow things down, and as we have more physical realism, it'll slow things down. That said, it could be very worthwhile, and in my lab, we have actually started working now with some virtual reality setups, so being able to just put on a pair of goggles and see everything in 3D would be fantastic. Right now, we're mostly doing it for visualization, but in principle, there's no reason that this shouldn't scale fairly comfortably to 3 dimensions.
So let's just say 2D, but you introduce a surface or something where now adhesion becomes important, that cells are more likely to survive if, say, they're connected to a surface versus just being
Absolutely. So
naked in the ocean.
That's one of the directions that we want to explore for why they would group to begin with for those sorts of abilities.
so much
Thank
again.
you.